don't forget, science is communication. Science is not only programming with your computer, it's always communication between you and I, so ask questions. Why do we have to compare 3D structures? We want to predict protein function somehow. Why do we need shape? So structure determines function. True. But why not go for it directly?
Because you don't have the same resolution to see like fine details, because function is always generalized.
What do you mean by function is generalized? That's a very generalized statement, Paul.
Yeah. Because
I'm sorry.
Function is always like enzyme or membrane transport or something like that. I mean,
membrane protein.
Yeah, membrane protein. Or you can always get specific, more specific and more specific. But if you then have something new where you want to predict its function, then you need to go back.
Can we not put a number on function? Can we not just, so one thing clearly, one answer that evolves from what you're saying is clearly true. 3D structure is something that we can easily measure. We can compare, we can manipulate in a computer. But we could map function onto a number, right? And then we could compute these numbers.
Yeah, but it's always taken from raw data, which we always start with the structure and go to the function, so if you want to compare. So if you have
no, but the raw data, oh, you say that the raw data for function, we don't have raw data? Yes, we do. We have the interaction, we have all kinds of aspects of it. Yeah, but it's an interesting point that that's an argument I didn't think about. This is an argument that you can actually build. You can put the argument up that the raw data for structure is much simpler in the sense that one experiment, even if it's a big one, so big one as defined as it's expensive and takes a long time. Nevertheless, it's essentially one experiment that gives you the three-dimensional coordinates of the residues, and that's it. While function is many experiments, and you have to, so it binds RNA, it binds DNA, it's part of a particular pathway. All of this is only different aspects of function, and none of these experiments that sort of nails one issue is nailing the whole thing. In that sense, clearly this is one way of defining function is more complex because it requires more data sets. It's an interesting definition. It's certainly true. And ultimately, another way of looking at it here that is a repetition of somehow the same thing is looking at one simple feature, one simple aspect of the simplicity of comparing structures with or sequences. And that simple is the issue of metric. When we have protein A, protein B, and protein C, be it as it is now, you can clearly say that protein A is similar to protein B. You can state that protein B is similar to protein C. Right, these are expressed here. But you do not have a validity of the triangular inequality. We can actually not say, this does not imply that C and A are similar. And that ultimately relates to the domain issue. And the domain issue ultimately relates to something like this one. We have one part of the protein that does something, in this particular case here, it's beta-stranded part, and then there's sort of an alpha helical part, and there are two different parts of the same protein. They're two different, in this particular case, they're not really domains, but domain-like subfragments. And what we could imagine is that evolution exchanges a block like this packed sheet, like this extended alpha helix with the two kinks in there. Because this extended helix with the two kinks in there binds DNA. And this is why you may suspect that you will see that again. And in fact, you do. So this is like an evolutionary module that you can put onto different things. So evolution moves in terms of these three-dimensional objects. And that is why, in fact, there is sort of something that you can recognize on that, and why, in fact, there is not a metric. Now, you can actually use even the sequence level in a simple way, and that has been published in a tiny paper here by my friend Zippel. And by defining a new metric that essentially defines the entire overlap of the entire protein, you can bring sequence comparisons to obeying a metric. It's sort of a little paper and a little trick, but it has an immense impact on database comparison. And actually, so this is Manfred Zippel here, and actually it is absolutely relevant if you want to sort of compare 3D structures on a large scale, 3D structures on the scale of proteins interacting with one another as one structure, and opposed to proteins that are sort of binding internally and seeing similarities on that level. in order to do these complex superpositions in 3D, the metric issue had to be solved first. And ultimately, it's only sticking it together. Now, there is this major issue of nomenclature. And let's just begin with a three-dimension structure. Three-dimension is very simple. You have an object that exists in a 3D space. You need 3 coordinates in order to describe the coordinates of the atoms of a protein, or even the residues of a protein. Very clear. Now, this particular version here is, of course, a cartoon. In this cartoon, you have highlighted secondary structure, in this particular case, the beta strands. There is a little bit of a helix that is actually here. It's hardly visible. That's 3D. Now I can take the 3D information and project it onto 2D. 2D distance map, shown here. Amino acids, or the protein running from the first residue to the 99th residue, first to the 99th. Anytime you see a black point here, that black point indicates that these 2 residues are near each other. Okay? That's a distance map. Now, you have reduced the dimensionality by 1. You can go 1 dimension further in terms of reduction, get to 1D. It's a string, it's a sequence. Okay? And it's just something that you can write as a string. Observe that in the column here, what I put is the secondary structure. E for extended. The blank here means neither helix nor strand, and I use only 3 symbols: the H helix, E extended, so the beta strand, and blank for nothing, for visibility. So you can actually write secondary structure as a string, because essentially it's a 1D feature. Okay? People sort of misuse in Wikipedia and in many books have sort of in their head secondary sounds like 2, and 2 sounds like 2D. Completely misleading. Secondary structure in terms of information content is 1D. Okay, let's go into the idea of what we lose and gain. When we go from here to here, we lose 1 dimension. Can we go back? So can we take a complete distance map without any additional information and infer the 3-dimensional structure from it? We can. How come you all know that? It's true. Your answer is absolutely right. But why? Why do you know?
Because we did it as freshman before this. Using going from a contact map to 3D structure.
Oh, cool. There's one problem with that issue. Do you know that?
I mean,
there's a sort of pseudo problem. It's not really a problem. You cannot distinguish the image and its mirror image.
Right.
So that's one which you can typically recognize on a protein when you look at it, which way, whether it's wrong.
You do this with a bit of precision, I would think.
That is the other issue. So in this transition, if I gave you more time on this image here, you may recognize that this image is not symmetric, and this is something that happened over time. Simply so this image at some point was in my PhD thesis. And over time this slide changed, so my PhD thesis is more than 1 year ago. And over time things changed, and the image suddenly is no longer symmetric. So if you lose that much information, so where do you see that's asymmetric? For instance, here this is much denser than there, right? So there are a couple of points you can, in a better projection you can actually see. So if I lose information in the transition from 3D to 2D, I cannot go back. True. But essentially, if I do have the full information, I can. Now, do you happen to know where this is relevant in terms of an experimental technique? Did you discuss that in the lecture too?
Why the contact maps are relevant? Well, because you can create contact maps a lot easier than...
That is a different story. So that is the story about our lecture. That's about protein prediction. But my question is, there is an experimental technique that essentially builds upon the fact that you can go from here to there.
NMR?
Yes, exactly. Because in NMR what you get is essentially distances. You get constraints for things that are close to one another. And you go from here to here. NMR is actually much more challenging because you do not get actually the color for I and J. You get the color between a particular cysteine and all the other cysteines. You do not know which cysteine, and that brings, in fact, the process of getting from an NMR structure to a three-dimensional structure is therefore one where you sort of create a model and then you measure again. So to exclude, you label certain cysteines, or you go step by step. And that is why that actually, it builds upon this idea, it builds upon the fact that you can go from here to here. The methods that do NMR, or you go from NMR constraints to structures, essentially are methods that do exactly this problem, convert 2D to 3D, except for they have additional problems. Now, we lose another dimension. We go from 2D to 1D. Can we go back?
Proteins are mRNA, they only have the sequence and they go to proteins. So
yes.
The information needs to be in the sequence. So
yes. Can we go back? Do you define yourself? Do you define yourself?
No, but
so the question is, what is you? If you define yourself as your proteins, and you say that in every single cell, at every single time moment, so many million proteins are doing that, so I can do it, then that's a fair answer. It's not really a practical answer, because you cannot make your proteins do that on my screen, so to speak. And on my screen, we can actually not do it. Okay, the information is there, but we cannot really go in terms of physics from 1D sequence, although we know that the information is completely there. We should be able to do that. We still, as a field of science, have not been able to do that. So there are approaches that are now much, much better than 15 years ago. And a lot of that has to do with the power of computers. So the brute force things the brute force solutions, ultimately the various generations of machines that started with the name Anton in the Institute of David Shaw. So they were special purpose machines that do molecular dynamics simulations on a huge scale. And they really have pushed this field of predicting protein structure from sequence. But where we are at is we can now do something like 50 residues, 60 residues in some cases, okay? Which is amazing. But it is not, many proteins are longer. So we still cannot quite do the entire protein problem with that. And there's a variety of reasons for that. So upon 1D information loss in that direction from 3D to 2D, you still have all the information there. From 2D to 1D, you completely lose it. Because you lose the context, and that is essential. Okay, and again, Paul, you're right. So many people now try, so the AlphaFold of Google, essentially the way it does it, it tries to sort of get secondary structures, solvent accessibility, and features such, and angles, backbone angles, predict the contact map, and then go to 3D structure. And ultimately the means by which they go from these deep learning predictions of contacts to 3D structures are exactly these NMR tools. These tools used to do construct NMR, to go from NMR data to 3D structures. So let's talk about sequence alignments. Ultimately, alignments answer the question how similar proteins are in terms of just letter comparison or string comparison. We would have these two strings, we would compare how similar they are moving around. We would see there's a certain similarity. We would bring in, so this is the pairwise comparison. This is what you call a multiple alignment when you go from 2 to 3. And so then you sort of move things around and you get a certain set of similarities. Now, we get now to the point, what we establish here is the similarity of strings, right? But the similarity of strings still, what we don't have is meaning. So one thing that we do know is this one. Or many people in the room will sort of know that this has a certain meaning. Some people will sort of recognize that this has a different meaning. And then you may recognize, or some in the room at least sort of, that is more complicated, but some in the room will clearly recognize the meaning of grandfather here. How many would recognize the meaning grandfather here? How many speak French? Papou? So there could have been a candidate here. Okay, so, but there is some similarity, and there's another father in the game. In some other languages, this is father. Now, so ultimately the issue is to go from the level of comparison of strings to the level of meaning. To keep some out, to group some, and to group some in a way that is reasonable. So ideally what you would want is that grandfathers are together and fathers are together, and things that are not belonging there are kept out. For the first step of our task, however, I will ignore that problem of meaning, and I will really go to the simplest problem, which is we define some objective function, and in a simpler way the objective function is identity of letters, and then we simply find the optimal solution. As I said here, percentage sequence identity. And ultimately, we are in the string matching problem. What we're trying to say is we ignore that the residue I has any influence of the residues left and right of it. Okay, we align every single column independently. And the string matching problem, essentially, let's begin here, is to simply move things around such that I find a good hit. If you do that, in this particular case, you would, for instance, see that the best way of matching these two strings here is one where you introduce two dots, insertions. What's the meaning of insertions? Or gaps is another way in which they are referred to. What does it mean? You have all had the idea of alignments, you have all had the idea of insertions and deletions. What does it mean? What's the interpretation of an insertion?
That maybe we can still show that the sequences are similar, but in terms of evolution there happened a deletion, for example, or something like that.
Does evolution delete the residue?
No, but...
Yes, actually it does.
Okay.
So it actually can happen, yes.
That was correct.
But typically, when I push you, does not always mean that this is suddenly the different answer. The idea of pushing is that you fall on the right side of the alternative decision. But it typically processes, in fact, that you add something. So, sort of cutting on one side is always putting another one in on the other side, right? Now, adding. How do you imagine adding in evolution? What does it lead to? Adding a residue. Yeah?
I think it depends on where you add it.
Yes, that is true. But let's look at the simplest idea. So in the simplest idea, and I'm now again looking with my hands, it always works not very well, but I never find objects that sit around that I can just pretend to be proteins. Well, it really doesn't work. I'm really not fit today. So those are the two things. And now say this one is longer, and you immediately see what happens, right? You have sort of an additional loop. You have an additional bump. And this additional bump may in fact be a new binding site. It may be that you sort of get a sensor out that gets a little bit further out, that you evolve a positive charge in there. There are all kinds of things in which you can imagine that. And this is in fact the sort of underlying model, that you have an additional embellishment. You see whether it buys you an advantage. Anyway, so the interpretation, the biological interpretation of these alignments is we have an evolutionary relation. Why do we care about an evolutionary relation? So it's fun to study evolution, that's true. But other than that, why do we care? Why as you all know, alignment is a very central issue. Is evolution as central? Well, it is, but in everyday sort of life, we're not caring that much about evolution. Why do we care about evolution in this sense? Maybe it's sort of giving it away already.
Well, if you know something about an evolutionarily related protein, then you can maybe imply or pull information from that, something where you don't know.
Why? Yes, that's true.
Because what you're saying is like that little bump isn't going to change the function of the entire protein. It may just give it a little bit more, take a little bit away.
But why is that the case? So if I let's go a step back. Let's assume that what we are comparing here are not objects from biology. But those would be, bless you, those would be randomly created protein sequences. So first of all, the reality is they have different features, okay? It's not a priori clear. We have to look at that, and there's a variety of ways in which you can show that in fact they have different features. It's not trivial. Makes a lot of sense once you have observed that thing. So they are sort of different things. But would the same still be true? Would it be as relevant?
If they had a certain percent of similarity?
Is it different?
If that 89% or 98%, something like that?
Sure. So my question now to you is, if at 80, whatever number is, at T, at the threshold T, so I have two proteins that have T residues identical. Or the fraction of their identical residues is T. The PID, I will use this word very often in my lecture, percentage pairwise sequence identity, PID. So say that is T, 60% or 80% or 90%, whatever that number is. My question to you now, does it matter whether I have some evolutionary connection between this pair or not? Is it relevant? Yes, it is. It's absolutely relevant, and that's why evolution is important. And the relevance ultimately comes from the fact that we assume I could in the computer find pairs of where you get a T, certainly not for the 99%, I'll give you that, Paul. So for the example that you constructed, your answer is absolutely right. But if I pushed, so I moved you in a very mean behavior of a teacher, moved you into accepting T independent of a number. The moment you put T as an independent number and to put that number to 10, for instance, or 20, at that point actually, whether or not they're related, evolutionary makes all the difference. If they are related, then the probability that they agree in the positions that are relevant for function is much higher than if you randomly created that overlap. So that is why the evolutionary selection system underneath is such an important piece of information. That's why we absolutely care. Evolution is not just something in our heads. It's something that is absolutely essential underneath in order to benefit from the data of sequence comparisons. Again, on the level on which you talk about, Paul, 90-something, that is not true. But as soon as we get into lower levels, and I'll show you the kinds of things that we typically do on those levels, this becomes very, very relevant. And we do essentially see similarity in structure and similarity in function of sort of diverged sequences because of the evolutionary connection. Because when things change, they typically are neutral, because that is an advantage. Or they buy us a new function. That typically is what we refer to as homology, and homology historically has a slightly different origin. Originally, it comes that's too long. The simple Greek background here really is from same homo is the same logos word, the same meaning, the same word, the same element, sameness, and it goes back to Owen, 1853, Richard Owen, two organs. So we're looking at the whale. The whale is a mammal, and you cannot immediately see where the whale has legs and arms. Is a an animal with two arms and two legs, and you're not seeing them, but the homologous organs in the whale, that is what this term has been used for. So there are sort of the corresponding organs. Now, in our everyday life of genes and proteins, we really go from the level of organs and the level of similarity of organisms to the level of genes, and we essentially say that two homologs are proteins that are similar, that are related in evolution. There are two different terms here, orthologs and paralogs. So orthologs are those that in fact happened before the speciation events, and paralogs happened from a duplication event. So paralogs essentially are proteins where you have another copy of the same protein. That's the idea, okay? So some people use the term homolog structure. This is then beginning to become really, really misleading. What we're talking about is similarity in structure. But underneath is important to have the evolutionary model, at least the moment t moves a little bit further down. There clearly underneath is the evolutionary connection. So let's go into the brute force solution, dynamic programming. Visually what you do is you simply take your sequence and you move it through until you find the optimal fit. And that's dynamic programming, going back to Needleman and Wunsch, published 1970, where you simply build up the matrix, protein 1, protein 2, and you look at all the possible matches that you have. You trace for the alignment building two letters, you trace the sum that you get, and you sum up over the diagonal, and this gives you a certain sum. Now, this certain sum, originally we don't allow for gaps. When you do allow for gaps, then you can increase the number of matches here. Allowing for matches is allowing for moves in this diagonal. And then you find an optimal solution. The optimal solution, this is a sort of trace, a dot plot, so-called dot plot, where you see that there are many sub-local optima. So the situation that I construct here is very simplistic. In a real dot plot it looks a little bit more complicated. There are alternative alignments, but typically there still is one best solution. Now in this best solution you always need to, the gap is a price you pay. So you get an advantage for matching identical amino acid, and then you pay some price for opening a gap. Now that means that in the simple model these two situations would be completely equal. Are they? Is one of them, so if you had in an alignment against this or against that, would that be similar? Or is there one that you would say is more likely to be right? Yeah?
And the left one is likely to be right because
yes.
it's possible that there is a long area.
There are two reasons. Very good, Kyla. So the one thing is if you already have a blob, a longer blob is more likely than many very short blobs. The other way that we can see when we see alignments is that is organism 1, 2, 3, and so forth. You see that there are some regions where they all align and some regions where they align here. On Tuesday we talked about domains. We would say this is evidence for a domain 1, domain 2, and then something in between where some organisms simply don't have anything. So that essentially is a gap for all kind of region, right? And this is what you see are blocks. How do you create blocks in terms of this objective function, where this is in fact the positive you get from hitting the same residue, and this is sort of the price you pay for inserting a gap. But in this language, these two would be the same. How can we make them not the same anymore?
Then a new parameter for extending a gap is...
And that is also first one?
No, it's both basics in...
I think albuminosity.
Okay.
Gap open, gap close.
Okay. So typically, where exactly you put this, gap open, gap extension penalty, there are different sort of things. 10 times is just one way of doing it. Then there is also allowing local alignments. That is this actual Temple-Smith and Michael Waterman algorithm from 1981. The final sort of dynamic programming that we all are aware of. Then comes this issue, what is best, local or global alignments? I'll get back to that in a minute. And the question is really the number of hits best? Well, clearly it's not. We already sort of talked about some proteins have very different lengths. So the number of hits clearly is not the best, but there is still this issue. Would it be best to have an alignment that matches two amino acids completely right? So essentially 2 and 2 right? Or would it be better to have 4? So this one is 4. Why do I have 4? On the next slide there's something went wrong. Where is the 3 from? So on the next slide something is wrong. I dropped an E. That's where it went wrong. Okay, so here's 3 and 7 because I made a mistake. Yeah, I made a mistake with the insertion here. I shifted the E. So I put the gap here instead of here. That's the mistake I made. So this of course should read 3 and 7, but let's just assume I had this right. So I had the dot differently, and then in fact the question really is it better to have 2 and 2 or 4 and 7? What's the better alignment? That's the kind of problem you open the can for the moment you say, I drop from the idea of a global alignment where I really do an end-to-end alignment, and I allow, this is Smith-Waterman, this is what everybody is using these days essentially. We all know local is the right solution, but we have by ourselves a problem. How can we solve that? Yeah?
Maybe if you would extend, I mean the 2 and 2 is pretty good because it's like 100% of what we have.
You cannot do better.
If we would extend the sequence and would get like 2 out of 7.
You do see what happens if you extend it. That is the alternative. You're not getting 2 out.
Then the 2 out of 2 doesn't become 2 out of 2, it becomes 2 out of 9 or something.
Yeah, but then you say you stop the alignment, right? This is exactly the idea of not a global alignment. This is the idea of a local alignment. That you say, well, I cannot align the sequence very long because the similarity is only there. But my question is, when do you say yes or no? When do you say I cannot do it or I can do it? Is 4 out of 7 acceptable? Okay, the immediate answer is more than 50%. But my question really here is, what is the better number?
We still need something that's like biologically relevant and just 2 amino acids.
How do you do that?
Yeah, well, I don't know. Well, 2 amino
Yeah, but
acids.
you're on the right. Why not? Why is 2? So how could you simulate biological relevance?
It's nothing. It's just it's.
Why?
Because through pure.
Sounds good.
chance.
That's the point.
I mean, yeah.
That's highly non-trivial. That's exactly the idea. Chance, random. You cannot really go to. My favorite biologists tell me what is reasonable. You need to question the data, query the data. And the way to do that is, what is random? So, which of these you would, for instance, could ask, which of these is more surprising with respect to random? Matching, if you look at a database of 150 million proteins, each of the most sort of 500 residues long, you see that and you have 20 amino acids, 20 by 20 is 400. So every pair of 400 will be in this huge space of 10 billion, right? This is absolutely clear. Even a triplet. So even if you went up to 8,000, still 8,000 in 10 billion is still a tiny number. Every triplet will be there completely random. And that is the point in which before you start with biology, before you sort of contact the biologist, go into your data and essentially random. It's not reasonable because random. Right? That's the point that you have to sort of get to. Now the next issue here is one that pertains to this to identity. I said I will use the term percentage sequence identity. That gets us to a slide, and that slide comes from Marco Punta. When you align these 2 proteins, both of those are potassium channels. One is in a eukaryote and one is in a prokaryote. In that alignment, what you see is, this is one of them, this is the other one. And the column in between highlights whenever there's a letter, it's the same letter between the 2. Whenever there's a plus sign, that means by some criterion they are similar. What is highlighted and the overall situation here is we have 26 of 72, so 36% identical, which is a sizable number. But most relevant is here that the potassium channel, or the recognition that in fact the bacterial one could solve as a model for the eukaryotic one, came exactly from this alignment, and it came from looking at the most important part, which is actually the recognition of potassium filter of the channel. And that's exactly in yellow here. In the yellow part you see one of the 3 residues in yellow is the same. One of them is sort of hydrophobic, both of them are hydrophobic, so they are related. But in one case here, really there is no relation. It's an alanine and an isoleucine. So there's absolutely no biophysical similarity. Nevertheless, the experimentalists who sort of were genius enough to recognize that this in fact would have a similar function, that meant that they could do the structure of this one, and that led to a Nobel Prize. That was the first potassium channel 3D structure. Now, ultimately, this entire idea of similarity goes back to Margaret Dayhoff, and Margaret Dayhoff simply in 78 had a large set of 71 protein families. I cannot remember how many sequences she had, with 1,500 variants between them, and that was sort of, she built the first protein atlas from this set of sequences. She essentially aligned them, superposed them manually. You see these dates are predating at least the Smith and Waterman algorithm. So essentially she did it manually, and then she essentially built these PUM matrices that count for differences and sort of compute the probability of random changes or changes in her data. A little bit later came the same idea from the Hennicoffs, Stephen and Jorge Hennicoff, simply later with many more proteins, and ultimately underneath is this block idea. So you build a log-odd matrix of proteins, pairs, where you simply say that whatever I align that I know is similar proteins, wherever they change, it's neutral, because they sort of do the same thing. They have a similar structure, they have a similar function, hence wherever they change is not really relevant. And I built a log-odd matrix from that. That's the Blossom 62 matrix. And there are a few things I want to point out too. First of all, the implication that you're moving from a letter identity to similarity is that the diagonal is no longer always the same. So mapping a W, a tryptophan on a tryptophan is much, 8 times more important than matching an A on an A, alanine on alanine, right? So this is 17, that is 2. Okay, that's the first observation. The second observation is that in some cases, even here we have an L on an M, leucine on a methionine, and that gives you a 4. So this mismatch gives a score that is higher than alanine on alanine. Okay, so mismatches can give you a higher score. And then the last point to make. Scores, mismatches can also actually count or pay or be negative. Okay, so the average of this matrix is chosen that in a typical situation the entropy of that is slightly positive. Today, many more matrices exist. Most methods somehow still use some derivative of the Blossom matrices, and that is very remarkable, because I should have had this. Do you know the story about the mistake? So the Blossom matrix was published in this paper that I quoted before, here, and there was a mistake in that paper. And about sometime in the 2000 something, somebody discovered that mistake. And they corrected that mistake. So people had been using the Blossom, most used the Blossom matrix with the mistake. Okay? So now people changed the mistake and did the right thing. What happened? The matrix is worse. We actually don't understand why. So the one with the mistake does better than the one where the math was done right. Anyway, dynamic programming gives you the optimal solution given an objective function, but there's a problem with time. The time used is in the order of the length squared. So essentially, okay, we have two proteins that are 1,000 residues long. 1,000 times 1,000 is 10 by the 6 computations. Why is that a problem? Why do I say this is a problem? 10 by the 6, these things, yeah? The phone can do that.
You have thousands of proteins with each other.
That's the point. I really wanted to, you were too fast, Paul, I wanted to water. So, I briefly brought up this slide before. When you look at the increase of the number of sequences, that isn't just as much as the computers increase. But the real problem here really is the number of comparisons. Yeah?
But we have to compare each protein against each other, right? Like single comparisons. Can we do that just parallel and then go faster?
It doesn't change it. That doesn't change the numbers, right? So the way computers, they do not double in speed every 2 years anymore. But the way they double is by getting parallel. So in this answer, or in this statement that computers double now every I can't remember what the number now is. It's more than 2 years. So double in speed more than every 2 years. That takes into account that you paralyze. So that's already, so if you could, and in fact this one you can optimally parallelize. You can boringly parallelize because you can actually run it on essentially different searches. So it's totally trivial to parallelize it. But the problem is that is already penciled in. So no, this problem of 20-fold increase over 2 years, the only way to address that is by tricks. And we are going to go through hacks. And we're going to go through some of these hacks before. There is a bunch of issues about parameters, such as the gap penalty and blah, blah, blah. But the main thing really is how to speed it up, and that's by hashing. Fast and dirty. Oops. Oh, I'm going in the wrong direction here. The way you do it, you begin with words. Say the word size is 3, you find 3 mirrors that match. In this particular case you would have 5 initial seeds that match, and then you would extend the seeds to either side. And then there would be something that you simply cannot do in the alignment. Now, in the extension of the seed to either side, you would use dynamic programming. And that is exactly what Psyblast, for instance, or let's just go, maybe I go back to that point later. Historically, the idea behind hashing came from William Pearson, with a method called FastA, which is essentially, you see, fast alignment. And then a little bit later came, or 5 years later, came BLAST, and the main person behind BLAST, Stephen Altschul, here, a team. And by the way, does anybody sort of recognize anything? So these are two competing ways of doing the initial fast alignment. Is there anything you see on these papers?
We both have Lippmann.
Yes. And science is a lot about communication. It's also a lot about knowing people. Communication is networking, and when you quote work, always quote the group the work comes from. You say, this has been quoted, this comes from Hager, et al. And Lippmann. Because most people know Lippmann. Do you know anything about Lippmann? So he's the NCBI, or has been the director of NCBI for many years. And he is on many other things. So Lippmann really is an outstanding scientist, has been an outstanding scientist and a very outstanding director. And he was, in fact, one of the people on two very competing approaches, so to speak. Does anybody know anybody else from these people? Altschul is the father of BLAST, Psyblast, and all of that, so clearly a known person. Is there anybody else that you have seen before or know anything about? So Webb Miller is behind a team that sequenced the first woolly mammoth, for instance, which I found remarkable, because the idea always was, for me, I always ask myself, why did they die out, what happened, and how similar are they to elephants, and that is exactly what was behind that sequencing project. Eugene Myers is the one who assembled the human genome at Celera. Eugene Myers, you do know, is currently Max Planck Institute director in Dresden, does all kinds of things, including sort of imaging the entire fly brain while it's sort of thinking. But you all have used a program from Gene Myers, I would argue, grep. It's also Gene Myers. So you do know sometimes people, be aware who they are. Anyway, going back to that issue. BLAST matches these fast words, and then it today extends in the direction left and right with dynamic programming. Why is it still faster? It uses, so it begins with a word, okay, and then it extends with dynamic programming. So it uses dynamic programming. How can it gain in speed? Yeah?
Divide and conquer, maybe.
Yes.
Separate the problem in multiple small.
Why is that so important? Because the time of dynamic programming is proportional to the square of the length. So by cutting it somewhere, say you cut somewhere in the middle, you immediately already 4 times, you go down in speed 4 times, right? But these 5 cuts that you have here, you immediately see how much you reduce, right? So extending here is much less expensive because you have short parts that you have to align. And that is why divide and conquer in this particular case works so brilliantly. because you're sort of beating something that in fact suffers from the fact that it's square in the length, and you cut the length. That's the brilliant part of it. The problem of it is, what's the problem of that? And that ultimately is the part where FastA was in fact doing better. FastA did not really survive time because it was slower. And was slower because it was better at the major challenge. What is the major challenge of this?
How do you connect the individual parts?
No, this is essentially, I would argue, the part where you can work, you can use dynamic programming. But the major part is actually somewhere else.
Maybe, I'm not sure, but maybe some sort of global references, because if
What do you mean by
parts
global
switch
reference?
to other parts of the sequence that are not close to the...
Oh yeah, that is a problem. Oh, so repetitions and all these kind of things. Or essentially, a linear comparison is always assuming the linear part. And you cannot sort of see that two regions are simultaneously similar. So if you have duplication of the same region in the protein, the repetition or these kind of things. Or you have an inversion somewhere, you read the genome the other way around, you insert that as an exon that is incorrect. All of these kind of things are difficult. But any kind of method, I would argue, has a problem with that. That's not what I mean. But what is the intrinsic problem that comes from this sort of dissection into short words? Yeah, I was about to say it's the Paul answer, yeah? Yeah?
It's not that you miss, that's what I meant actually. It's maybe if you separate them into shorter problems, you can't see the big picture anymore when you...
Yes, but it's already a big picture. I'm not sure whether you were saying this, would say this. You also put up your hand. Were you about to say the same thing as before? No, okay. You should have said the same thing as before. Essentially random, right? So the chance. By chance you didn't do the right thing this time. So the major issue really is to have words that are random hits. Okay? So we need, the entire idea is there is a background probability of match, and there's an actual signal of match, and we need to have the actual signal far away from the background. For that, we need the background. And that is in fact the big difference between FastA was that FastA essentially created the background on the flight. It simulated the sequences, while BLAST guesses the background from the amino acid composition of your data set. So that is one of the reasons. So why when you find a similarity between two proteins with BLAST, and whether or not the second one is found in a small database or in a large database gives you a different score, because by larger or smaller you make it more significant that you have the same distance from the middle of the distribution, from the average of the distribution here. Okay? Because if you are a certain distance away from a huge distribution, it's more significant, a huge database distribution, than when you're at the same distance away from a small database distribution. So this is important because you have the same alignment and you have different e-values. Okay? And that really is because ultimately you steer the estimate here simply by the size of the database and by the composition of the database you use. The amino acid composition. With that, BLAST guesses this background. That's why it can be so fast, because it does not have to simulate sequences. It just assumes the background. It works extremely well. How accurate are pairwise alignments? I'm going to go through a couple of things, but let's begin. This is the percentage sequence identity. So one thing is clear. There's a zone where sequence alignments are trivial. Therefore I call it daylight. So you have enough light to make a decision for any pairwise alignment. Okay? Then we get into a zone that was called twilight zone by Rost. And ultimately the idea simply is you lose the signal. Okay? So some of the hits you find in that region are right and some are wrong. And then there's sort of a midnight zone, the inverse of that. So essentially you no longer have a signal at all. Okay? So now we want to sort of see how the transition from one to the other is going to look. And what I'm going to show you is some percentages or more likely something like this. So I want to see when I come from 100% sequence identity going down to somehow into the twilight zone, back into the midnight zone, how are the number of pairs that have similar structure increasing in blue? How are the pairs of proteins that have different structures in red are increasing? This is fake data. It's not real data. I'm just throwing something. I want to make a plot like that. I expect something like that. I expect that there is some growth of blue and red. I expect that at some point the red are growing more than the blue. I have to expect that because there's a daylight zone in which the red is 0. That's the definition of daylight zone. I don't make a mistake, right? So that's why the red has to at some point, wherever that point is, right? But somehow the red, how these curves look, I don't know. But essentially the basic fact that both curves somehow will increase and that the red will sort of begin to touch the blue at some point is what I expect. How can I create the data for that? Again, you could Google and you could find papers that do this for you. But if you were the first, and it's not that long ago, on my time scale, 20 years ago or something like that, people had not really done that. How would you do this? What would you need?
You need the ground truth.
Yes, but what is the ground truth you're going to use? So if I had to give you something, what would you ask me to, if that would be an exercise, what would you want me to give you? And what would you do with it?
A large amount of proteins where the structures are already known.
Yes.
I can compare them all to another, group them based on how their similarity is. And then
Which similarity?
Pardon?
Which similarity?
Alignment similarity.
So sequence.
Sequence similarity.
And
then, if we wanted to create this one for the twilight zone, you could basically look at all of those that had then a sequence similarity of like 20,
yes.
15, and 18, and 17, and see how many false positives and true positives are there.
Where does true positives and false positives? Where do you get that from? I don't know what that means.
And
can somebody else fill in? So essentially he's on the right track. So you need a data set of proteins. For that data set of proteins you need some level for sequence similarity. And then there's something about true and false. That part I didn't understand. And so there are some simple scores such as root mean square deviation. This is in fact what I show. So you take the protein of known structure and you do a 3D comparison of all of them. You take the same set and you do a 1D comparison, sequence alignment of them. And then you're going to get labels of the type 2 angstrom here. If they are less than 2 angstrom RMSD, you say they have a similar structure. So that ends up being the blue ones before. If they are less than 5 angstrom or 8 maybe, this is our data today, I would use 8 or 10. We say they are red, they're clearly not related. And then there's a realm in between here where I cannot really say. Some of those may have similar structures, some have not. I will stay away from that. And I will just create 2 very extreme data sets. Red, blue, okay? Now, it's not quite possible that way. We cannot align PDB all against all because there's a lot of bias in the data set. The bias comes from the fact that I have 150,000 proteins in the PDB in that ballpark. If I would just remove all the proteins that have 100% sequence identity to each other, how much do you believe would be left? So I take my protein 1 in the PDB in my data set, in my list. Then I take protein 2 and I ask, is protein 2 100% identical to 1? If so, drop it. If not, take it. How much of PDB would be left in this simple algorithm? Ballpark. Any idea? Yeah?
80%.
No, it's fair. It's an absolutely fair number, and it's less than half. But yes, why could you imagine that you would have the same thing twice? So if it's less than 50%, then we have at least 2 versions of the same thing on average. Why could that be the case? Yeah?
Do you mean like biology-wise or?
Whatever you want to say.
PTMs, post-translation.
For instance. So ultimately, PTMs is already, yes, ultimately this is a particular, is a very specific answer to that question. But much more generally, whether protein binds or not, you still want to know whether the structure is changed through binding. The protein is the same, right? The protein sequence is 100% identical. You measure it with and without binding. And that is in fact something that you absolutely want to have. You want to see how the binding changes the protein structure, right? This is exactly how you study a function. So a lot of the PD will be the kind of things that in terms of what we want to do here is just bias. Very, very important for the biology, for us, not good. Because it would give a very, very wrong image if somebody studied the particular binding of one particular molecule many, many thousands of times. And there are, in fact, many in the PDB where that is the case. So what you need to do is you need to cut out a sequence-unique subset. So a sequence-unique subset essentially means that no pair in this data set here is similar beyond some sort of level of sequence identity, okay? And then we take this sequence-unique subset out and compare it against the rest. Now we cannot do sequence unique against sequence unique because there is in that set no similarity that is sort of significant. So if you do this against itself, most of what we want to look at is gone. Okay, and the first two somehow did that were Chrysander, who is now at Harvard, and Ryan and Schneider. And what they found was this plot, the HSSP plot, number of residues aligned, percentage sequence identity, and curve means that everything above that curve has a similar 3D structure. There was this minor changes when you had a larger data set here in gray, and that is essentially everything above this gray line has a similar structure. The raw data, similar structure blue, different structure red. You see that there is some red above, but it's difficult to see that this is actually not, in terms of numbers, not very high. I'll show you the number in a minute. And a lot of the blue here is up here. There's some blue down here. But above, you find blue. Examples from this plot here, you have a case of these two proteins here that have 35% sequence identity at 150 residues. So they are sitting up here. Here's a case where you have 40%, 50 residues. And they are sort of sitting below the curve, or just below the old curve, so above the new one. So they both would be deemed similar, essentially. Okay, now we're going to essentially look at a value that looks at difference to this curve here. So difference to the curve itself is 0, and then we're going to have plus 5, plus 10, minus 5, minus 10, and so forth. And that's going to be on the x-axis. And on the y-axis, I'm going to show you here the number of pairs of proteins that have a similar structure. You see that's the 0 number, the 0 value, and that's plus 10, minus 10. Logarithmic scale of the number. The important observation here is that if you call this curve, or this point here, this sort of the middle of the twilight zone, or some part of the twilight zone, you see that upon, or in this twilight zone, not clear now where you define the twilight zone, but this somehow is a zone here where you see that the ones you want to find increase by a factor of 10. So you go from some number here, plus 5, to a number that is more than 10-fold higher at minus 5. Okay? Within 10 points, so essentially 10 percentage points of sequence identity, a lot is happening. Now, when you look at the negatives in the same, so the negatives somewhere are 0, as I said. The definition of the curve is that there's a little red above it. There is some red above it. But you do see that the negatives grow by 5 orders of magnitude over the course of this. Or let's just go for this point here. We are about 5 times 5,000 here. Go to a number of 1 million. Okay? And that means that we go from a point where most of the hits you find are blue to a point where the vast majority of the things you find are not correct. And this happens, again, within in a spread of 10 percentage points. So this is essentially what physicists would call a phase transition. So you have a dramatic change upon the intrusion into this twilight zone. Now you can ask, why is that so? And there is some evidence that there is a phase transition. And the phase transition, the way to sort of rationalize it is when you can always sort of find a residue to be neutral in change. And you can, if you had that one, then you can find another one that is neutral, another one, another. And at some point you get to a point where you can no longer sort of account for that, or make the step that prevents you from falling, so to speak. And then you suddenly fall. And that, in some sense, is what we believe we see here. Anyway, that's what we see. Now, is there anything else that we can do other than pairwise? And I'm going to go over that one. But clearly, on the level of multiple alignments, we should get more information than pairwise. Dynamic programming on multiple is complicated simply because of the time issue. There is the claim, and again I don't have a quote for that, somebody told me that they, and it's a few years ago when this was still a number that sort of impressed you, if you have a machine with 60 terabyte of memory, today most of you running around with your computers, you're still impressed by 60 terabytes. We, in our group, we would still like to have a machine with 60 terabytes. But today this is easy, this is doable, right? But they went up to 6 sequences, right? What is a typical family of a protein that we want to align in multiple sequence alignments today? Is it 6,000s, in fact, today? Yes, many thousands. So there are these methods that you use for evolutionary couplings to do alignments, and for those 50,000 is better than 10,000 as a signal. So many thousand. So 6 is not really relevant, so essentially whatever hack, and again here clever hacks from David Lippman, going back to David Lippman, to do dynamic programming pairwise, only space in the vicinity of intersection, totally clever. The dumb approach that originally Rost proposed is much simpler. To map the problem to the tree, to map the problem to pairwise comparison. And here essentially is the visualization of that problem. You have 4 proteins you want to do in a multiple alignment of these 4. You first begin all pairwise alignments, that gives you this matrix here, and then you simply pick the pair that is most similar. Say A and B, 90. And in step 1 you align A and B. And in the alignment of A and B, you're going to come up with a consensus sequence that takes whatever residue they have. But if both have the same, you take that residue. If they are different, you take the residue, whichever residue has the highest information content. Okay? According to the Blossom matrix, for instance. In the second step, you could take the next 2, do the same thing, and create another consensus sequence. And then in the third step, you would have a consensus of the consensus sequence. So you would have easily mapped the problem of aligning 4 sequences into 3 pairwise comparisons, right? It's a hack. There are different ways of doing that. You could sort of align the next one into this one, and there are many ways of doing that. But ultimately, this hack is still with us today, and that allows you to, multiple alignments allow you to go deeper into the twilight zone and do much more. Now,